As with all labs, you want to make sure that you have your printed out procedural guide with any notes that you write when you're watching the video that you're watching right now. So make sure you have that annotated set of instructions. And what we're going to do now is we're going to go ahead and prepare the Direct M slide. So for that we need a slide, right? And this I cleaned with soapy water, dried it off with the lens paper. You need your swab, so you can swab the inside of your mouth. And you need the Sharpie, right? So those are all the things we need right now, although we'll need this other set of items in just a moment. So let's go ahead and start preparing our slide. And so for the label here, just as it says, and what I'm going to do is I'm going to avoid putting my fingers down where I'm going to put my sample. So I'm just going to go ahead and put this down, and I'm going to put 1 DIRM for direct mouth. Okay, so that's what we've got there. And just as long as you know what it is, you'll be good to go. You want to go ahead and let that dry, or while it's drying on the other side, remember, so what you need to do is you need to take it where it says 1 Direct M, and then go ahead and flip it over. And on this side, you're going to draw 2 circles, just like you did for your previous slides. And the marker is on the bottom side so that when you put your sample on the top, the marker will not be there. Okay, so now what we've got is this is labeled 1 Direct M on this side, and on the other side, you've got the 2 circles. Okay, so I can go ahead and put that down now. Now let's go ahead and swab from the mouth. So the purpose of this is to pull off your human cells. So I'm going to be pulling off some Andy cells. And along with that, some bacterial biofilm and some bacteria. So don't get too aggressive with it, but you do want to move it along the inside of your cheek to pull off that, as well as around your gum line perhaps. Now for this procedure, you shouldn't do it right after you brush your teeth because you've removed all of what we're trying to get at. So please, and eating food right before you do it also doesn't help because then you're just going to have a bunch of nasty food particles in it, right? So you want your mouth to have gained its natural microbiota flora back. So, you know, wait a few hours after you brushed your teeth to let things go. So I'm swirling it around, getting the gums. Okay, and now down on the slide, what we're going to do is we're going to go ahead and swab it. And we did 2 circles instead of 1. Flip it over, swab it. We did 2 circles instead of 1 like the original directions said, and that's so that if one of them doesn't come out too well, then you've got the other one as a backup. So go ahead and swab it. And what I see right now, let's see, is yeah, you can see that. See how it's flashing? There's like gunk there. That's the polysaccharide. That's the mucus that we all have in our mouth, which is really important. And that is where a bunch of cells are going to be at. And so what we're going to do now is I'm going to go ahead and put this swab back in here, and I'm just going to throw this away. It was just in my mouth, you know, no worries there. And then what I'm going to do is I'm going to let this slide dry for about 10 minutes. Now it'll take longer to dry than some other samples because that mucus, that polysaccharide will hold onto water more readily, so it takes longer for it to evaporate. So let's go ahead and wait no shorter than 10 minutes before we go to the next step. So I'll see you then. So we've waited a little bit more than 10 minutes. So our slide can now be heat fixed. So again, go ahead and put your clothespin on it. And essentially, remember, it's really important to have the material on the top, right? So this is now facing straight up. So when we look at this, we read that it says 1 Direct M here, right? And that means that the circles are on the bottom and the swab material is on the top. And if you look at this, if you look at it from the angle, like right now with my eye, I can see that the material is on the top. So for the heat fix, remember, you want to stay not really close to the bottom of the flame, but towards the upper. And it's a pretty quick 1, 2, 3. Okay, so what we're going to do now is we're going to go ahead and let that cool down a little bit. right? Because, well, when we add the stain to it, we do not want it to bubble and burst and such. And so from that perspective, let's talk about what we're actually doing here while this is cooling. So the heat fix has two purposes. You may recall what those are. One is to fix the bacteria to the slide, get to stick to the slide, and to essentially fix all of the external elements of the bacteria in place. So it is to kill the bacteria and to stick it onto the slide. If you heat fix too long, you keep it in the flame too long, or the real hot part, then what will happen is the sample will either get incinerated, just like when you're putting your loop in the flame, right? And it turns red and essentially turns everything into carbon dioxide and other compounds. Or if it's too liquidy, right? If you didn't wait the 10 minutes and you did it, then what you may see is that all the samples are destroyed because it boiled the samples. So don't do that. That's why these steps are timed. That's why we do things certain ways. So make sure you pay attention to those details. So now let's go ahead and do the staining. And for this, what we need is we need our little staining tray. We need our crystal violet because we're only going to do the direct stain right now. I've got paper towel for the blotting. And then I've got this water right here for rinsing. Okay, and this simple stain is called simple for a reason. So let's go ahead and do this. So for the simple stain, what you want to do is put your sample on the tray. Go ahead and shake up your crystal violet, right, number 1. And then go ahead and essentially put a drop or two and so right now it's not staying in the circle. It's kind of going all over the place. That's why this clip is purple to begin with, because it's been used before. Okay? So basically we're now going to wait about 60 seconds. It can be longer for this to penetrate the cell itself. And since it's at an angle, it's going to want to curve. So I'm just going to actually just hold it here while I do this. Or I can go ahead and just kind of rest it there. Okay? The directions actually have you put the slide all the way in the bottom without your, so you could do that. But I like holding on to it with this. All right? So basically, you know, being a microbiologist means being patient. So now that we've had enough time go by, you're going to put it at an angle, and you're going to go ahead and take your distilled water, go ahead and rinse. Right? Now, if you see, there's purple at the bottom. So what we'll do is we'll go ahead and take our paper towel, we'll put it in the tray, we'll go ahead and let it actually pull. You see how it just pulled the stain there? So let's go ahead and add a little bit more. And now we see that there's still purple that is being pulled off. What you want to do is you want to do this until you don't have that purple coming off anymore. Okay, the purple that is coming off. So now if you look at this, right, so now we have colorless liquid. Oh, maybe one more time. There we go. And we go ahead and put it down like that. And so the liquid now, the water, is colorless, which means that all of the crystal violet that is not bound to the cell is now off of the slide. Okay, so now what we're going to do is we're going to go ahead and blot this. Okay, and it's called blotting for a reason. We're going to go ahead and put it in there. And I'm just going to go ahead and pat it like this. Go ahead and do that again. Now you're not pressing down real hard because you don't want the material to come off, but you do need to press down on it. Remember, the bacteria in your human cells should have stuck to the slide if you did this properly. Okay, so that's what we're going to do. Nice and easy. And now if we look at this, what we can see is there is purple material, right, on the slide. You can see that through the white background a little bit. It may just look dark to you, but there's little speckles all over the place, and that is areas of high concentration of stained material. And what we may see when we look under the microscope is areas where there's not obvious to our eye purple, there still may be some cellular material there because cells are really small. Okay, so that's what we've got. So this slide is now ready for, I'm looking at this because I'm wondering if my choice of paper towel was a bad one, because what we don't want is we don't want a whole bunch of fibers all over the place, right? So what I may need to do, and we'll check this out when we actually do the microscopy, if we need to adjust our slides a little bit, rinse it off again and blot it with something else, I'll do that. But this will be a good example where when we look at it under the microscope, if there are fibers from the tissue, then we can look at that together, right? Identify what it is, and then we'll go ahead and alleviate that problem right then and there. Okay, so at this point, this slide is now ready to be viewed under the microscope, so we're not going to do anything with that right now. So what we can do is we can put it in the slide box, except for the fact that we don't have any room because we already put 3 slides that we made from the previous exercise that we are going to do gram staining on, right? So what I'm going to do is I'm just going to take out one of the ones, for example, amoeba whole mount. Since this actually has a cover slip on it, you don't need to protect it as much, right? The bacteria in your human cells are right on top, and so basically that's the one that's more important. So I'm going to put that in there, and what you can do is perhaps just take your amoeba slide. No, maybe not. Some slide boxes you can put 2 in one slot, but not this one. So this amoeba one I'm just going to put in a drawer, and when it comes time to do the microscopy, I'll pull it out. And I'm putting it in an area where it's not going to break, but again, the slides that you make are more precious in terms of how you have to go ahead and protect them, protect the tops of them. Okay, so what I'm going to do now is I'm going to go right into the gram staining for the slides that we already prepared. But what we're going to do is we're going to go ahead and conclude this video, and we'll pick it up in just a moment.